SBGrid weekly webinar series. Today we've got Rubén Sánchez Garcia, and he's going to be talking to us today about DeepEMhancer, which is a post-processing tool for cryo-EM map sharpening. If you have questions during the presentation, please send them to the hosts in the chat. And our webinar next week at the same time is going to be Fabian Schlofeld, who's going to be talking about using 2D class averages or computing 2D class averages in GPU Isaac. So with that, I will thank you all for joining in. Thanks, Rubén, for being here to present, and I will turn it over to you.
Well, thank you very much for the invitation. It's a pleasure to be here, and thank you all for your interest in our tool, DeepEMhancer. So in typical cryo-EM workflows, we have to collect thousands of movies, and from them we gather even millions of particles, with the objective of refine a 3D volume for the protein complex we want to study. But in order to get the most information from the map, we really want to get atomic structures, atomic models, and this model building process is complicated, time-consuming, and to be honest, it is very difficult, or if not impossible at all, to build these atomic models directly from the refined volumes. Instead, you need to post-process them if you are in this side of the pipeline, or pre-process. It depends on which side you are, for two main reasons. The first one is that the reconstruct maps tend to exceed some noise, and although in many cases this is not a problem, for flexible proteins or for membrane proteins, this noise can mask the actual signal, and it is difficult to distinguish between the noise and the signal. And also, experimental maps, reconstructed maps, they suffer loss of contrast at high frequencies. And in order to deal with those problems, masking approaches and sharpening approaches are used. Masking is sometimes more an art than a science, and regarding sharpening, there are several different approaches. And there are practically every package has their own, and they are good for most situations, but there are some differences. And among all the different sharpening approaches, we really like this log scale approach. Why? Because this is a local approach, and it means that it deals well. It deals well with heterogeneity, and also because this is a knowledge-based approach. It makes use of atomic models. And what do we mean by that? Well, in order to run log scale, you need to have as input both a cryo-EM map and an atomic model for this map. And if the map, if the model is perfectly fits to the map, if this is a perfect model, you will get ideal scaling factors, and the sharpening, the local sharpening, would be perfect. So in best world scenario, this is kind of a gold standard sharpening, but it has a strong requirement to meet. You need a good atomic model for your input map. And this is something that you, in many cases, you don't have, or even if you are dealing with a novel protein, you may not even have a single atomic model at all. So what we wanted was to get a similar post-process map, but without the need of this input atomic model, just using the cryo-EM map. And of course, as you may imagine, skipping this part, it comes at a cost. It is not free. And the way we pay for it is just learning from data, from the structural databases, from the PDB and the ENDB. We learn how does cryo-EM maps and atomic models relate. And this is DeepEMhancer. And the way we made it was basically we compile a data set of experimental maps and atomic model basis, and we run this log scale on it. And also we compute high quality masks from the atomic models in a systematic way. And as a result, we got these masked log scale processed maps, which look very nice in most situations. And now what we did was to train a neural network in order to obtain this kind of results. And the good thing about this approach is that all these things, this log scale, this atomic model requirement, all these things you only need to do once in your life, or better said, we did it for you, so you don't have to care. When you are using DeepEMhancer, the only thing you need as input is the raw map, and it will produce a high quality post-process map version. So if you are interested into the details, you want to look under the hood how it works, well, you can go to the bioarchive manuscript. It's been since like the beginning of summer. It's been out, but for those of you that just want to give it a try, I guess that that this is a good starting point. And this is a tweet that was published in September, and in which basically there is a guy that said that DeepEMhancer works well for his case, and you can see that it is the case indeed. You can see that there is much gaining in details for this particular case. And since in those days, if somebody in Twitter says something good about something you have done, I think this is a very good sign of that this is worth it to try. And just a few other examples, just to give you an impression of which kind of things you can get. If you compare this original map with the one that can be obtained with DeepEMhancer, you can see how this distal part is well more defined than in the original map, and also some tricky parts for instance this loop that has not originally modeled. This loop becomes more clear, and indeed you are able to trace this part of the loop thanks to these enhancements. And this is just my final example from another publication in which you can see how DeepEMhancer has been able to separate better the two chains of the two chains of the protein, so that you can model it better. Okay, so let's say you want to use it, then you need to know that there are two ways to run it. The first one is a command line tool, which is a Python 3 package, by the way, or you can also run it within a Cypion version 3 as an XML protocol. And if you want some help to use it, if you want to know how to use it, the first thing I would do if I were you would be only to ask the program for help, either as the command line -H option or just clicking on the help buttons in the Cypion XML protocol. If you have some doubts, some comments, some questions, you just can go to the GitHub page and open an issue, and I will try to solve it as possible. There is some documentation over here. If you need something else, I would try to do my best. For sure, you may spot a bug, so this is also the good place to report bugs. And if you are using the Cypion version and you get some problem using it on the Cypion, I would recommend you to contact directly the Cypion team using their email address. Okay, so how do we use DeepEMhancer? Well, in the command line, you just have to provide an input and output volume. And in XMIP, you just only have, in Cypion XMIP, you only have to provide an input volume and just click on the execute button. And if you're using the command line, you need to provide inputs as MRC format, whereas in Cypion, it will deal automatically with the format. So choose whatever option you prefer. And that's all. For many cases, I mean, and from average user, I guess that the default parameter, default options would produce a very decent job. And this is because DeepEMhancer is a fully automatic method. And by fully automatic, we mean that it only needs the input volume. That's all it needs. And this is a major advantage compared to other methods. I'm gonna discuss slightly more in detail which are the most important options. So the first one is the input. The input could be either a mask, either a single volume or the two half maps. And in principle, those options are equivalent. They produce the same quality results, but it is important to notice that the input should be unmasked and unsharpened. This should be a raw map, the map you obtain directly from reconstruction. This is how an input map should look like. We need the noise, either as a spherical noise cell or as a cube of noise. But DeepEMhancer needs this noise in order to get the best results. So, and it will work by default using these parameters, and it will produce good results providing you have these noisy volumes. So probably one of the most important parameters for DeepEMhancer is the input normalization. And we have two different ways to normalize the map. We have the default normalization and the mask-based normalization. And why is it important? Because when we were training the neural network, the different maps that we were using, that we collected from the databases, they had different scales of intensity. So before processing them, we needed to normalize, to standardize them, their intensities. And the way we do is in the default option, we just take into account the mean and standard deviation of the noise, and we normalize as done in many other places, just taking the mean and dividing by the standard deviation. Whereas in the mask version, we normalize with respect to the protein mean and protein standard deviation. And in our experiments, we have obtained way better results using the default option. This mask option, it's a last resource that you may want to try if you only have the already masked volume. But if you have the original volume, please go to the default normalization, because there is some information in the noise. And if you remove the noise, if you get rid of the noise, then the neural network would not be able to perform as well. And here you can see an example of the default normalization and the mask normalizations. The result seems quite similar, but if you pay attention, you could see that there is slightly more details using the default normalization. But still, if the only thing you have is a already masked map, masked option is the only possible option. And this mask is only a binary mask, an MRC file, in which the protein part is set to 1 and the other parts are set to 0. Okay, so if we focus on the default normalization, DeepEMhancer would try to automatically compute the mean and the standard deviation. And in order to do so, it will compute a radial profile of your protein complex. So this works quite well for many cases, and especially if you are in the case that you are providing the whole cube full of noise. In those cases, it will work like 99.9% of the times. If you're providing radial noise, it will work in many cases, but it may fail for other cases. And the kind of pathological things are either hollow things such as viruses or elongated things. For instance, in this virus, if you compute the radial profile here, you can see that there is a very thin layer of signal, and all the rest of the cube is noise. So in those cases, DeepEMhancer may fail estimating the mean standard deviation. Or for elongated fibers, since they are not spherical but cylinders, a radial profile is not the best way to determine it. However, in those cases, what you can do is just to compute manually the mean and the standard deviation of the noise. And the way I tend to do is only in Chimera. I just open the volume in Chimera. I tend to crop a region of the map that contains only noise, and then I ask Chimera for the statistic of the volume. which are basically the mean and the standard deviation. And this is the parameters you have to provide to DeepEMhancer if you want to perform the manual default normalization. And the second very important parameter that you can select in DeepEMhancer is the model power. And by default, it uses the type target. If you don't set any parameters, it will use the type target in the command line. And this is the first selected in the Scipion protocol. And in order to understand the differences, we need again to go to the workflow. And all the three options, they employ the same kind of neural network, but the difference is how they were trained, the training set we used. So if we distinguish the high-res model from the default and the weight model, this high-res model was trained on high-resolution data, only high-resolution data, whereas the other two were trained on a wider range of resolutions. What it means is that this high-res model tends to perform slightly better when you want to use it on high resolution maps, very high resolution maps. But the other still should perform well in the case of high resolution. And when we compare the white target mode, we have these three: white, target, tight, and high. So when we compare this white with the other two, the only difference is that when we produce the inputs for the training, the references, sorry, we employed a wider mask, whereas the other used a very tight mask. So what you would expect from this model is to obtain volumes that are not as masked as the others. They are wider, they are softer, and the boosting, the enhancement is smaller. But there are some situations in which you prefer a softer transformation, because otherwise you may be losing some part of your protein or something like that. You might be masking out some parts of your volume that you think they are actually protein, but the neural network thinks they are actually noise. So in those cases, this white target is the best option. And again, if we go to this example, we can compare for this particular example, which is very challenging because it has like a lot of membrane over here. So you can see that the tight target is the model that it's able to remove most of the signal coming from the membrane, and it produces very high resolution details over here. The high-res model is able to produce slightly more high-resolution details, but it comes at the cost that it has not been able to remove completely the noise. And on the other hand, the white target, what it generally does is to perform a very poor masking, and you can see here that even the membrane is not masked out compared to the others, and that the details tend to be wider over here, but still the results are pretty good. So which option should you be using? Well, this is my advice. I guess that the tight target gives good results in many cases. So I think it's the best place to start with. But if you have a very, very good resolution map and you are especially interested in enhancing only the parts which are very high resolution, this model would work the best for sure. And this white target is your last resource if the other two options do not produce the results you are expecting, or if you realize that there are some parts of the protein that have been masked out, which is very unlikely, but it may happen. And this is my piece of advice. In the end, you probably want to try both, and you want to trace your protein using the both maps at the same time, and you want to model the different parts using the different results. And also, you may like to add a third map produced with other tool like local deblur, for instance, in order to compare the different results and try to assist you, because for any different protein, the best method would be different, and in some cases, just comparing a few different post-processing a lot.
Rubén,
yeah.
one quick clarification question about the model power. Is the distinction just the training set that was used for parameterizing the model, or are there algorithmic differences too?
No, they are exactly the same. The only difference is the target we want to recover. In the high-res, it was aimed to recover like the very high resolution features, whereas in the other ones, the training set, it covers a wider range of resolutions. So it should be more generic. And the white and tight difference, it only refers to how masked the input should be. That's all. But
Thank
the
you.
method is exactly the same.
Great, thank you.
Thank you. So what I was saying is that in this case, what I would probably be doing is to compute these two maps. And for the central part and the upper part, I would probably be paying more attention to the high-res model. But when trying to model the transmembrane part, I would try to pay more attention to the tight model. This is my advice. It depends on the protein. In many cases, you would get almost exactly results, but it may be some difference in some examples like this one. And let's just discuss another few options. So we have like two computation options. You can use it on GPUs or CPUs. GPUs are pretty fast. You can be dealing with a map in 15 minutes or so. So this is even faster than the original log scale in many cases if you are using a GPU. And you can use more GPUs at the same time. So it scales linearly with the number of GPUs. And the batch size is another parameter that may help you in some situations. The batch size, it's the number of cubes of the protein that are simultaneously processed, of the volume that are simultaneously processed. It has no impact on the quality of the results, but only in the computation. And basically, if you get some out of memory error, some CUDA error, TensorFlow error, CUDNN error, whatever, and you don't understand why, the first thing you should try is to set this batch size to the minimum possible value, which is 1. And if it does not work, then we have a problem. But if it works with the smaller batch size, the error message only meant that you were out of memory, that you needed bigger GPUs or that there were other processes using the GPUs, so you cannot compute simultaneously 8 cubes. But if it is able to run with 1 cube, that's fine. It will take more time, but it will produce the same results. And the bigger this number, the faster the results would be. And the last option is just a classical image processing option. This is equivalent to the high dust option in Chimera, just to remove those spots of noise that are not connected to the main component. And this is I don't particularly like this option because I think that in 99% of the situations, the DeepEMhancer is able to mask out the noise almost perfectly. But in some cases, some people would like to enable this option. And finally, I can, since this is a Python package, you can programmatically make use of it. You can just import this class, which is responsible for the post-processing. And in a few lines of code, you could be performing the same post-processing as in the command line, and very simple. And it will take care of the normalization and all the details. So if you want to integrate this tool in others, this should be fairly easy. And that's all. I want to thank the people from Biocomputing Unity in Madrid, and also the people from the Vargas Lab, because this project was initially developed in a joint project between both labs. And I hope this has been useful. I know it's been very short, but this is the good thing about fully automatic stuff. You don't need to care a lot, or even at all. And I will be very happy to take questions.
Thanks, Rubén. That was a very interesting talk, and we do have some questions coming in. So again, for folks who, if you have questions, please feel free to send them in the chat. And if you'd like to unmute yourself, let me know. Otherwise, I will moderate them and collate them and go on asking. So I guess one question regarding the training set. So are there issues with proteins with things like metal centers or heme centers where the density inside the protein region may not be standard protein density?
There are some examples in the training set that contain different ligands. So the short answer is that we cannot know in advance if it will work perfectly for those cases, but what we have observed is that thanks to these ligands and other things that are not protein, but they are still contained in the dataset, DeepEMhancer tends not to remove them. But we cannot guarantee that, but in most cases, the density is still preserved. And this is one of the reasons I always recommend to use several maps at the same time. But it can deal with ligands. Yeah, and even some post-translational modifications such as glycans, they are not removed in many cases. They are still over there. And sometimes they are even enhanced.
Okay, thank you. So I guess moving along with the list of questions, we have one from David Hasselbach. David, do you want me to ask that, or you should be able to unmute yourself if you'd like to ask?
Yes, hi. Great talk, great software. So the Twitter example that you showed from seemed to have a lot of anisotropy, and I'm wondering whether, like, is that something usual that you can solve, and how much of like preferred orientation or anisotropy in the map could you solve, and is there specific settings that you would recommend to?
No, so far. We are working in a new version, but the current version is able to deal with only moderate amounts of preferential orientation. And the main reason for this software doing that is that it works as an encoder-decoder approach, in the sense that you have your input, you compress it into an abstract representation, and then you expand it back. So since in the expansion path you only have like actual maps, like non-anisotropic maps, I mean, this decoder approach is only able to produce non-anisotropic maps. So providing your input is not encoded too far away from where it should be, it will be able to deal with this anisotropy. But so far, we haven't systematically studied which are the limits. This is something we have to do. We are still working on trying to get better understanding of how to deal with this.
Thank you.
Thanks. Next, we have Jean-Paul Amarache, and I apologize if I'm mispronouncing your name.
Don't worry, it's Jean-Paul Armache.
Jean-Paul Armache, thank you.
Yeah, hi. I mean, wonderful implementation. It works very fast, and I'm always amazed by what it can produce, right? So of course, there's always this question about how well you work on your cryo-EM reconstruction, right? You have beautiful features and so on, to the level where once when I saw a map, I thought, holy wow, magic, right? But when you're publishing something, right, there is a possibility that, for instance, reviewers will look at this, because there is this perception that deep learning and so on is in a way black box, so you're not quite sure what you're applying and so on. So can you potentially share your point of view on how how the post-processed maps using DeepEMhancer are perceived by the wider EM community, and whether you can always trust the output of this, because I validate this using other maps, right? I do I apply different sharpening ways. I filter maps at very different levels, etc. Right? So I have a sort of a feel of what DeepEMhancer actually did and so on. But that's not something I can always potentially present to the reviewers. And sorry for this long-ish kind of.
And I think this is a very important, very interesting question. And I think that validation in general, it's a very important. And we shouldn't, we should take into account that there are biases everywhere in our workflows. If we are using templates, there is a bias there. If we are using initial models, there is also a template there. There is also a bias there. So this kind of neural networks, what they do is to use to produce some output based on a bias, and this bias comes from the corpus, from the knowledge contained in the databases. So as you are using bias, one way or another bias, there is always some risk. But the other option is not doing anything. So there is a trade-off we have to leverage. And this is my personal view. But I think that the way we should think of these DeepEMhancer approaches and other similar is not as the ground truth, but as a human expert, or like a human expert, an expert that has been exposed to hundreds, thousands of different cases, and it is a good advisor. It will tell you, okay, I think that this part of the protein is not noise, and I think that this part is noise, so I'm going to remove it. But this is nothing but an advice. And I think that at that moment, it's when validation should come, and you should try to validate probably at the atomic model building step. Probably this is the best place you can try to validate things, but you cannot know in advance if the results are correct. Although we have done an extensive benchmarking, we haven't found any potential issues, but there should be this disclaimer that says it may fail in some particular cases, some pathological cases. And you should be on charge. You are the pilot, the one that has to take the decision. On the other hand, this is like a hammer. You can break a head or nail, right, with a hammer. So I think that it can save a lot of time when you are tracing a protein, because you will go straight to the main chain, to the backbone. You can see directly the side chains. And once you have made your model, go to validate with the original map, because you don't want this to go straight to the end. You just want it to help you building models. This is my main take home message. Validation is an issue not only for deep learning, but for any other method that uses some bias. Yeah, I agree with you. Some reviews may not like it. My advice is use several methods so that you have like a consensus, and consensus tend to be way more powerful always. And in the end, you have to validate as much as you can.
Thank you.
So I guess jumping into one question that seems highly related to that is, are there any issues using the modified maps for model map FSC? And if I'm interpreting what you said right, it sounds like you would not recommend using that as a correlation between the model and the modified map.
And I mean, you can do it, but in the end, you need another alternative way of validation. You need yeah, because what this method does is to compute, let's say, a very smart filter. It computes a nonlinear filter, very complex, of your map. So it is producing a filtered version of your map, but you cannot know if this particular region was removed but should be there. You cannot know. Although I would say that this is very unlikely. We have never seen it, but in the end, you have to validate things. And this is also important that we stick it to a unit architecture, because we didn't want to have a generative model like a GAN or a variational autoencoder. And the reason is that these generative model, especially GANs, they tend to produce higher resolution images, for instance, in real world scenario. but it is also true that they can produce more artifacts. So we prefer a lower enhancement, a lower resolution enhancement approach, but that was less prone to produce artifacts. This was our approach.
Thank you. I guess moving down the list of questions, so we have Yuri Iozo, if you'd like to unmute yourself and ask.
Hi, Rubén. Very much, very interesting. I'm using it a lot already, produces indeed amazing results. I have two short questions here. So one, because I didn't dive deep into the algorithm, how it works. Is any difference between using one map or two half map? Is the first question.
There is very, very, very little, providing the whole map, the full map is not pre-processed. If you are using the map you are obtaining directly from refinement and there is noise in your map, you can see the cube or the sphere of noise, the results would be equivalent or almost equivalent.
And second question may be related to that. So I tried DeepEMhancer with the map output from Reliant and system. It works very nicely with the Reliant. But with system, every time I have problems, the DeepEMhancer says volume is hollow and gives an error and stop. I clearly can see that volume is not hollow, but maybe it's because system by default masks volume and I should generate, I didn't try that, maybe I should generate an unmasked volume.
Well, I guess that the unmasked volume should be your first option, because you know the best results that you can obtain with DeepEMhancer is when you have this whole cube full of noise. So if there is noise over there, results won't be of the same quality. But in the end, if you are obtaining this mask map, you could also try to use the map normalization option, the other option, and it should be giving some results.
So the hollow error is likely comes from that, right?
The hollow error comes for sure in the process of automatic noise estimation. Do you remember this file profile I said? Basically, it tries to find where the noise is automatically, and it was not able to find the noise. So you have two options. One option is to use the other normalization trick, which would produce results, although maybe not as good. And the other option is to compute manually the noise statistics. We can go to the slides 1 second. We can go over here. Yeah. So we have two kinds of normalization: default and the mask one. If your input is masked, there is no other option but to use this option, the mask option. But if your input is not masked, but still you are getting this hollow error or any other error at the beginning, you can just try to provide manually which are the mean and the standard deviations of the input. This is the way to deal with the error.
Thank you.
Basically, it tells you, hey, I've not been able to compute in a reliable manner which are the mean and the standard deviation.
Thanks, Rubén.
You're welcome.
Sorry about this, the slight delay. I'm going through and correlating questions. So I guess, but also one related to you were just talking about the masking, and you'd mentioned, this is a standard 1-0 binary mask. How would you recommend people handle cases where there's density that, we're pretty sure this is solvent, there's density that's pretty sure we're protein, and there's density where we just don't know. And in a binary mask, you've got to pick one.
Yeah, I would go tighter if you don't mind to lose these kind of regions that you're not sure about it. And if you have to pick one, I go tighter.
Okay. Let's say there have been projects I've worked on in the past where it's, the density that's poorly localized is unfortunately the density that's biologically significant. And that's
Yeah, I mean, for those cases, the best way is to try to use it automatically, or at least, I mean, in the default mode, because if it is able to see the noise, it will be able to learn to make use of that. Whereas if you are using this mask version, it is only able to see what the protein levels are, and this is not as powerful.
Thank you.
And manually computing the mean and standard deviation, I promise, is not that difficult. It takes 3 steps in Chimera, or you can do it in many other things. So if you just want to make sure that the noise estimation is perfect, providing you have noise, of course, do it manually. It takes 1 or 2 minutes.
Is there, I guess that's kind of a follow-up to that, is how do you know if the noise in your map is following the same, it looks like the noise in the training set. And I apologize if this is not going to be a fully coherent follow-up question, but
Well,
if there's something wrong with your data where experimentally it's just skewed in some way where the data set is broken, is that something that you're going to be able to detect with the noise comparison?
Well, first of all, the noise is compared internally by the network. The noise at this stage, it is only used in order to normalize the whole volume. But what we want is to scale the intensity values so that the proteins always look like in the range between 0.5 and 1.5 or so. So this is only for scaling. And providing your noise is consistent, this scaling should be working, because we don't really care about the noise, but the protein. Trying to learn what is noise and what is not, it is way more complicated than trying to learn what is protein and what is not. The only thing that we need the noise is just to make sure that the scale of the intensity values is the same that we use for training. But yeah, and there might be some pathological cases in which this does not work well. We are not aware of any, but I can imagine it that you have very nasty things that gives you a very bad normalization at the beginning. It won't work.
Yeah, well, I don't have a tremendous amount of experience with cryo-EM, but I could envision something like, okay, there's a pixel that's overloaded, and again, that's an analogy from X-ray stuff, or there's a uranium particle or a gold particle that we were using for registration.
Yeah, well,
that's now we have an outlier and our mean is off. But if you're calculating the density manually, you just avoid that region.
That's true.
So. Thank you. And again, I apologize for the poor coherence on that question. All right, and I don't see any more, so I think that may have covered all of our questions. So one last call, and if not, thanks everybody for attending, and thanks Rubén for presenting. It's a very interesting tool, and
thank you very much. It's been a pleasure.
Well, appreciate it, and stay safe and good luck. And
you too. Bye-bye.